Okay, this lecture is about pH, acids, excuse me, acids and bases. Here are the learning outcomes we're going to cover. So let's talk about what makes an acid and a base. What this diagram is showing you is pure water will spontaneously break apart. Okay? And water, which we know is H2O, will break apart into H plus and OH minus. You know, a lot of times H plus interacts with another water molecule and you get H3O plus. For the purpose of this class, we're going to use them interchangeably, and I'm going to focus on H plus. So in pure water, what I want you to see is that water will break apart and reform, and break apart and reform. And so the amount or concentration of H plus ions equals the amount or concentration of OH minus, right? It's just a water molecule breaking apart. And these two ions can contribute to something we call the pH level. Okay? And over here, you see a scale for pH that goes from 0 to 14, with 7 in the middle being neutral. So water is neutral if it doesn't have anything in it, right? And it goes back and forth between water and breaking down into the ions and forming back water. So that's why at neutral, the amount of H plus equals the amount of OH minus, and we get a pH of 7. And we're going to talk about these numbers in just a second. Okay, so that's one of the natural sources of acids and bases. There are other acids, like you've probably heard of HCl, hydrochloric acid. This is an acid that will fall apart into an H plus ion and a Cl minus ion. We have bases such as sodium hydroxide, which will break apart into a sodium ion and a hydroxyl ion. And so in biology, we have things that add acid and base to our systems. And at the end of this lecture, we're going to talk about how we control that, because you don't want to go to extremes of acidic or basic solutions, for instance, in your blood, or you will die. So let's talk about what is actually pH. So the definition of pH is the measurement of the hydrogen ion concentration in a solution. Okay, so the more hydrogen ions equals a more acidic pH, but the value of the pH is a lower number. And we'll explain why that is in just a second. So what I want you to do is look at the pH scale and notice from 0 to 7 is where it's acidic. Okay, so acidic acids have a pH of 0 to 7, and a base or a basic solution, let's say acidic, is a pH of, I'm going to put 7 in parentheses because we know that 7 is neutral, to 14. All right, so the lower the number, the more acidic. The higher the number, the more basic. And that's because, by definition, an acid is a molecule that donates H plus to a solution. Okay, so if you're putting in more H plus, you're going to be more acidic. That's what an acid does. A base reduces the H plus in a solution. So the less H plus you have, the more basic the solution is. So you need to know those definitions, and you need to understand this pH scale of low pH is acidic, high pH is basic. Another term for basic is alkaline. So you may have heard this term as well. An alkaline solution is a basic solution. All right, so what do these numbers mean? Okay, I don't want you to get bogged down in it. It's really simple as far as what we're going to do in biology. So let's look at this side first. This is how you calculate the pH of a solution. So the pH is the negative log of the concentration of H3O plus, which I told you we will just talk as H plus. So I'm just going to show you an example first. A pH of 10 is the negative log of 10 to the minus 10 molar H plus concentration. Okay. Molar is moles per liter. It's a way we calculate a concentration, so don't get bogged down in that. You'll work with that in chemistry. Let me give you another example before I do that. The pH of 2 is the negative log of 10 to the minus 2 molar hydrogen concentration. So I want you to take a minute and look at these crazy equations and see if you see an easy relationship. And that easy relationship is, I want you to see the pH number is the same as this exponent, just positive, right? So these are negative log 10 calculations. You really don't have to calculate. For this class, we're not going to make it complicated. We're going to keep our pHs as whole numbers. So if I said you had a pH of 6, what would you write in the concentration brackets? You should write 10 to the minus 6 molar. So you have to have your units. That's telling us concentration. You just take this 6 and you put it up in the exponent. And so if I said my hydrogen ion concentration in my solution was 10 to the minus 3 molar, what would be the pH? You would say the pH is 3. That's as in-depth as you need to go as far as calculating the pH. Right? So it's just this number as a negative exponent or this number as a positive whole number. Okay. So over here, it shows you that the concentration of hydrogen ions times the concentration of hydroxyl ions always equals 10 to the minus 14. This is where we get our pH scale. So at neutral pH, we have the same number of hydrogen ions and hydroxyl ions. That's by definition neutral. And so we'd have a similar, or the same, I should say, this 4 is bugging me, concentration. Okay. So at neutral, what's our pH? Our pH is 7. Okay? All right, let's look at the scale a little bit more. So again, the scale is going from 0 to 14. It can be written either way, 14 to 0, 0 to 14. This is just showing you some common solutions you might be aware of. Excuse me. So bleach, is very basic. It has less hydrogen ions. It has more OH ions. It's alkaline. Over here on the bottom part, we have coffee and urine and lemon juice, and look at the acid in your stomach. Very low pH, so there's a lot more hydrogen ions, less OH minus, this is for acidic. So why does that work, that the more hydrogen ions, the smaller the pH number? Okay. That's because pH of 2 is 10 to the minus 2 molar hydrogen ions, right? That's the concentration. And this number, if you wrote it as a decimal, is 0.01 molar. Okay. Now if I go down to pH 1, I'm at 10 to the minus 1 molar, which is the same as 0.1. Right? So look at this lower pH has a greater concentration of hydrogen ions than a higher pH. So pH of 10, which is 10 to the minus 10 molar hydrogen, is 0.1, 2, 3, 4, 5, 6, 7, 8, 9 hydrogen ions. Oops, let me put my molar. Right, this is a really small number. So the higher the pH, the smaller amount of hydrogen ions. The lower the pH, the more hydrogen ions. And it's because our molarity is at that negative exponent. All right, let's look at the pH scale again. So here it's upside down again. You always got to look at which direction. And what I want you to notice is this correlation. So an acidic pH, there's no decimal here, right? pH of 1, 10 to the minus 1. pH of 2, and in case this is confusing you, 10 to the minus 0 is 1. Remember, anything to the 0 power is 1, right? So as we get more basic, we're getting a smaller and smaller amount of hydrogen ions. The other thing I want you to notice from this scale is that these are 10 times difference, right? So pH of 3, 10 to the minus 3 ions, which is 0.001, compared to the pH 4, 10 to the minus 4, which is 0.0001, it's a tenfold difference. And as you increase the pH, you're decreasing. So pH 4 has 10 times less hydrogen ions than pH 3. This is a smaller number than this number, and it's smaller by tenfold. So if you went from pH 3 to pH 10, let's say, 7 pH values, that means you have 1, 2, 3, 4, 5, 6, 7. You have 10 million times less hydrogen ions than at pH 10, than pH 3. Right? 10-fold 7 times. So the difference is 10 to the 7th. So there's a 10 million fold difference. And as you increase the pH, you're going to decrease the hydrogen ions. So we'll try this in just a minute. All right, so why do we care about pH? So pH affects molecular interactions. And especially protein folding. So if you watch the macromolecules lecture, talk a little bit about the effect of pH on protein folding. We've used this for many, many years to preserve food. So vinegar is very acidic, and we call it pickling food, like pickles. And what it's doing is it's reducing the pH so much that proteins start to denature. They start to fall apart. They don't have their function anymore. So if you happen to have some contamination, like some mold or some bacteria in your pickling, it would all be dead because it's so acidic, the proteins fall apart, and proteins are essential for cell function. And so it's a way to preserve food. And when we say preserve food, what we're really meaning is, excuse me, eliminating contamination of things like bacteria and fungi that can normally live and replicate. Right? So, pH is interesting for that reason. pH is also important, if I could spell, in your blood. Your blood pH is somewhere between 7.35 and 7.45. That's a really tight range. And if you get too acidic or too basic, you can die. Mainly because you're interfering with molecular interactions. So what we have in our blood and other places are something called a buffer. And so a buffer is a chemical that helps minimize the change in pH. So a buffer can go back and forth to add or remove hydrogen ions based on what else is in the solution, and it helps minimize the change in pH. So it's going to keep things stable. So let's look at the buffer system in our body, in our blood. So we have a bicarbonate buffer system. Okay. We can regulate our pH, or we do regulate our pH long term, by breathing and by using our kidneys. So in your lungs, you normally breathe out CO2. In this equation, you can see that CO2 and water interact and make carbonic acid. Carbonic acid can donate a hydrogen ion, acidic, and make this bicarbonate ion. If you have too much acid in your system, the kidneys will get rid of it through your urine. So if you look on your scale here, urine, oh, that's quite a range. That's interesting. In general, urine is more acidic. So we're releasing, it all has to do with how hydrated you are, right? So if you're very dehydrated, you don't have a lot of extra water to get rid of, your urine's going to be very acidic. And if you are nicely hydrated, so when you pee, it's nice and clear, not dark yellow, then your urine will be more basic. But your urine is the way the kidneys excrete extra acid. Your lungs are the way we also help get rid of extra acid. So by releasing CO2 into the air. So this is a system. In the blue, it's showing you the same kind of thing. Here's your capillaries. They're going to take CO2 out of your cells, and it leaves through your lungs. And oxygen comes in, and we have this gas exchange, right? Gases go by simple diffusion, so they can just move right on out of cells when the CO2 concentration is too high. Here's just another diagram showing you that when carbon dioxide is exhaled, you're shifting the equation and also getting rid of some of that acid. What happens when someone hyperventilates, right? They breathe too fast. You don't get enough CO2 out. You're breathing instead of nice exhale to get the CO2 out. And so you build up CO2, which goes back, and we actually build up a base. Okay. So our system can get too basic. So if someone's hyperventilating, you have them breathe slowly, they're getting rid of more of that CO2, and they're helping restore the blood pH. So let's look at, let's just really quickly look at our outcomes before we do a couple questions. So the concept of pH is talking about this relationship between acids and bases in our body. To differentiate an acid from a base, you're going to be given a level of pH, right? So a base is greater than pH 7, and acid is less than pH 7. Right? This means that there's more hydrogen ions and this means that there's less hydrogen ions. Be able to calculate pH given the hydrogen ion concentration, right? So if I say there's 10 to the minus 4 molar H plus, then you know the pH is 4. And vice versa. If I tell you there's a pH of 4, you know that the hydrogen ion concentration is 10 to the negative 4 molar. And finally, the action of a buffer is just to help maintain that pH. We're not going to do any kind of calculations. I just want you to understand that the whole point is to maintain a pH level of whatever the cell or the blood or other solution needs. Right? Okay, so let's try a couple questions to see if you're getting this. All right. The first question is adding acid tends to blank of a solution. So take a second, figure it out, pause the video. So adding acid tends to, we know it increases the hydrogen ion concentration. That's the definition of an acid. But then the trick here is, remember, it lowers the pH value. So the more acidic, the smaller the pH value. Just look back at your pH scale. All right, if the pH of a lake is 4.0, what is the hydrogen ion concentration of the lake? So if it's pH equals 4, then you have 10 to the minus 4 molar hydrogen ion concentration. So that's the correct answer. You've got to have the unit on it. Okay, a solution at pH 6 contains blank, then the same amount of solution at pH 8. So how does the hydrogen ion concentration change between these two pH solutions? So we have 10 to the minus 6 compared to 10 to the minus 8. So we know, because 10 to the minus 7 is in the middle, we have a 10 times 10, we have 100 times difference, so it's C or D. 10 to the minus 6 is larger number than 10 to the minus 8. So the answer is C, 100 times more H plus. Okay, the last question. A person is taken into the emergency room after going unconscious due to alcohol. The person has low blood pH of 7.23. Remember, we want it to be somewhere between 7.35 and 7.45. That's your goal. Which of the following will improve the patient's condition? So you want to give them some base. You want to increase the pH. So you don't want to give them more acid. Obviously, you can't do all of the above. And breathing into a bag will increase the CO2, which will actually increase the pH. So breathing out CO2, will decrease. Breathing out CO2 will increase the pH, which would make it more basic. But if you're breathing into a paper bag, you're going to increase that amount of CO2 because you're kind of rebreathing, and that's going to make you more acidic. All right, so those are the information of pH and acids, bases, and buffers.